Hello students, welcome back to my channel CSIR Buddy, and in today's video we are going to study about composition of blood, and this is related to animal physiology. In the previous video I have already taught you about ABO blood grouping, so if you haven't watched that video, first watch that video. I will be providing the link for the same in the description box. So let's get started. So first of all, reading about the history of blood. At the beginning of the 20th century, an Australian scientist, Carl Landsteiner, noted that the RBCs of some individuals were agglutinated by the serum from other individuals. So, Carl Landsteiner noticed that the RBC of a particular person, when treated with the RBC of another person, so some of the through a serum, some of the RBCs or the blood, not RBC, blood was showing agglutination. And what does agglutination means? Agglutination means clumping of blood. So you can see like this type of clumping in a blood. So this is meant by the agglutination. So by this he concluded, he made a note of the patterns of agglutination. Now he was making a note about the patterns of how agglutination is working with particular serum, and showed that the blood could be divided into groups. And this is how he concluded that there are some of the groups in which a particular blood could be divided. And we already know in this time in modern science that the blood groups are A, B and O, or AB blood groups. This marked the discovery of the first blood group system, and that blood group system is known as ABO blood group system, which we are following till date. So now through Carl Landsteiner we know that there is a group system that is known as ABO group system, blood group system, and earned Landsteiner a Nobel Prize, and for this discovery he also got a Nobel Prize. What does agglutination means? That means clumping. Now moving further. Now the composition of blood. So blood has two things. One is plasma and other is corpuscles, and corpuscles are particular cells. Right? Plasma consists of 55% of total blood, and rest of the percentage is comprised of corpuscles. Now corpuscles are of three kinds: erythrocytes, that is RBCs, which are red blood cells; leukocytes, which are white blood cells; and platelets. So the function of RBC is mainly of transport of food or transport of oxygen and CO2. The main function of WBC is to protect against any disease or any bacteria or something, so it is helping in protection. And platelets are involved in blood clotting, right? So this is the basic composition of blood. So blood is composed of plasma, which is the liquid portion, and the corpuscles, which are the cells. So cells are of three kinds: WBC, RBCs and platelets. Now pH of blood is slightly alkaline. So slightly alkaline hota hai pH, that is 7.35 to 7.45. Now there are two colors of blood. One is bright red, and if a bright red, you see a bright red colored blood, then that means it is oxygenated. And what does oxygenated means? That it is consisting of O2. So O2, the RBC must have been carrying the O2 gas more. Now in dark red color, we notice that it is deoxygenated. That means oxygen is not present in this case. So therefore it is dark red. So one is bright red and one is dark red. So ये गाढ़ा दिखेगा, एक डार्क वाला और एक ब्राइट रेड दिखेगा, मतलब हल्का दिखेगा. कलर जो होगा ब्लड का. Now coming further, talking about the plasma. So plasma, as already I have told you, that it is liquid part of the blood, and 91.5% it consists of water. So there is large percentage of water. 8.5% solute, which is gases like CO2, O2, N2, that is carbon dioxide, oxygen, nitrogen, right, and other waste products like urine, uric acid, creatinine, bilirubin, and ammonia. So these are the solutes, and solutes constitute of 8.5%. Now 8.5% के अंदर और क्या है? इलेक्ट्रोलाइट्स, न्यूट्रिएंट्स, विटामिन्स एंड मिनरल्स एंड प्लाज्मा प्रोटीन्स. सो इलेक्ट्रोलाइट्स Na पॉजिटिव, Cl नेगेटिव, K पॉजिटिव, Mg² पॉजिटिव, Ca2 पॉजिटिव, SO4 नेगेटिव, एट्सेट्रा. Nutrients, amino acid, glucose, fats, glycerol, and then vitamins and minerals. Then plasma protein like albumin. Albumin is 54%, that is osmotic pressure maintained. So albumin is maintaining the osmotic pressure. Now globin 38%, which is used for as a defense mechanism, and fibrinogen, which is 7%, and it is used for clotting, blood clotting. So this was the composition of plasma. Now going further, hemopoiesis, blood element formation. Now when a blood is formed, that process is named as hemopoiesis. Now before birth, what happens is that in the yolk sac the blood is developed. So जब अभी baby हुआ नहीं है and it is yet developing, then the production of blood, the blood formation takes place through the embryo sac, and then liver, spleen, thymus or lymph node, right? As the baby turns into a 3-month baby, 3 months before birth, then the blood formation is done in red bone marrow, right? So till date the red blood is formed in the red bone marrow, but before these months, before the birth of the baby, the blood formation takes place in the yolk sac or liver, spleen, thymus or the lymph node. Now coming further, talking about RBCs. RBCs are the red blood cell, and we also name it as erythrocytes. RBCs production. So kidney erythropoietin stimulate RBC synthesis. So the RBC synthesis starts from the kidney, right? Hematocrit, percentage of total blood volume occupied by RBCs. So the hematocrit means कि कितना परसेंट RBC ने ले रखा है the total blood volume. So how much percentage of RBC is present in the total blood volume. So in the case of females, it is seen that there is 42% of RBCs. If there is 100% blood, so 42% is RBC. And in the case of males, 47%. So 47% is RBC and the rest is the other things. Then the normal level should be in females should be 5.4 billion per microliter. So this is microliter. And in the case of male, it should be 4.8 million per microliter. If low RBC count is there, this is known as depressed hematocrit, and this will cause anemia, that is loss of RBC. If high RBC, elevated hematocrit, so it will show elevated stage. So this condition is known as polycythemia. So in the case of low RBC, we are calling it as anemia, and in the case of high RBCs, we are calling this as polycythemia. So I think this is also clear to you all. Now coming further towards the structure of RBC, how the RBC is looking. So as we all know that RBCs when mature, they lose many organelles including the nucleus also. So nucleus is absent, and as the nucleus is absent, its appearance is like a disc shape. So it is जैसे दोनों तरफ से अगर न्यूक्लियस नहीं, न्यूक्लियस सेंटर में प्रेजेंट होता है, so it will be like compressed from the between, right? So it will appear like a disc shape. So this structure consists of biconcave. So this structure is known as biconcave structure, right? So RBC structure is a biconcave structure, have higher area and lack nucleus and other organelles. So when RBCs mature, then they lack other organelles as well as the nucleus. Diameter should be 7 to 8 micrometer. So micrometer diameter. RBC produce ATP anaerobically, no mitochondria, because of the absence of mitochondria. As we know that mitochondria is the powerhouse, and if there is absence of O2, it will respire anaerobically. But if O2 is present, then all the cycle goes, the electron transport chain takes place in the mitochondria. But in the case of RBC, we don't have this. The mitochondria is itself missing, so there is no chance of aerobic respiration. So therefore RBCs respire anaerobically and then produce ATP, right? One RBC will consist of 280 million HP molecule, and HP is the hemoglobin, right? Hemoglobin from which the oxygen or carbon dioxide binds. Now 12 to 16 grams of HP in every 100 ml of blood. So 12 to 15 grams of HP is present in 100 ml of blood, 100 milliliter, right? So you can also see this diagram of RBC blood cell diagram. Top view of the red blood cell, hemoglobin rich cytoplasm. So the cytoplasm is hemoglobin rich and the shape is biconcave shape. Biconcave shape. Why biconcave? Because the nucleus is absent. So I think this much is clear to you all. Moving further, now talking more about hemoglobin. So hemoglobin is a protein, and globin, that is 4 polypeptide. So this is majorly made of 4 polypeptide. That means 2 alpha unit and 2 beta unit. 2 beta unit constitute of 1 hemoglobin. As it consists of 4 subunits, so every subunit bind to each of the oxygen molecule. So that means that 1 HP can hold on 4 O2 molecules or carry on 4 O2 molecules. So 1 polypeptide. So polypeptide chain consists of 1 hemoglobin, 1 iron and 1 oxygen. 1 hemoglobin, as I already mentioned, that it will carry of 4 oxygen molecules. Now RBC has carbonic anhydrase. So it would convert O2 plus CO2 to H2CO3 and then HCO3 plus H positive. CO2 carried in RBC, CO2 is carried by the RBC, HCO3 dissolved in the plasma membrane. So this dissolves in the plasma membrane itself as carbonic acid, and then CO2 dissolved in the plasma, and CO2 dissolves in the plasma. Now RBC life is only 120 days. After that it has to be removed. And how it has to be removed? After destroying it, and the organs in which they can be destroyed is known as spleen, liver or bone marrow. So there are 3 organs in which the RBCs could be destroyed. One is spleen, another is liver, and then bone marrow. So RBC death and phagocytosis. So अभी RBC का death हुआ, phagocytosis हुआ. So it converts into heme and globin. So first of all, talk about the globin part. Globin part was the protein part. So as we all know that protein is made up of small chains of amino acids. So when we will break these protein molecules, then we will get amino acids. Now these amino acids are used for the synthesis of some other protein. Now the other part is the heme part. Heme, either it will follow this one pathway in which it is going towards the liver, or it will follow this one pathway. So heme would convert into ferritin in the liver itself. The ferritin is like Fe3 positive, would be carried by the transferrin. So it is carried by the transferrin, then erythropoiesis in bone marrow, and it will be destroyed in the bone marrow. Or what will happen, the biliverdin would convert into bilirubin. Bilirubin will, which is found in the liver itself. Bilirubin is a pigment, so you should know it is a pigment which is found in the liver. It will convert into bilirubin. Bilirubin will secrete into the SI, that is small intestine. So bilirubin is a pigment which gives the yellowish color to our excreta. So it is the one who gives the yellowish color. Now the bilirubin would be converted into urobilinogen, and then urobilinogen will be converted into stercobilin, and stercobilin would be released into the feces. And then if it goes into the kidney, then what will happen, it will convert into urobilin, and then it will form the urine. So this was all about the RBCs. I hope you enjoy this lecture and you like this lecture, and all the doubts regarding this topic is clear to you all. If there is any doubt anyways, you can ask me in the comment section. And if you like this channel, please like, share and subscribe to my channel, and please help me grow faster and faster. Thank you.